All right, I think we'll go ahead and kick things off. First and foremost, welcome to the Working with Contract Research Organizations and Contract Manufacturing Organizations to Accelerate Preclinical Development webinar series. I appreciate you all joining, and we're looking to have a wonderful time through this virtual format. My name is Chad Jackson. I'm the director of the Preclinical Translational Research Program at the Foundation Fighting Blindness. For nearly a half a century, the foundation has been focused on driving research that will lead to the most promising preventions and treatments and vision restoration therapies for a spectrum of degenerative retinal diseases. It is our pleasure to partner with Odylia Therapeutics to add another brick on this path to progress. This webinar, number 3, is part of a 4-part series installment for preclinical and translational research activities that we're all looking to learn more about. This series will focus on genetic technologies for blind diseases and will provide tangible advice and educational materials to researchers in the inherited retinal disease space, as well as the broader rare disease field. Before I turn things over to Dr. Ashley Winslow, the Chief Scientific Officer at Odylia Therapeutics, and our moderator of the session, I would like to review some logistical details of this session. Currently, all participant lines are on listen mode only. To ask questions during the session, please use the Q&A feature on the Zoom control bar to type in your question. If you would like to make a comment to the panelists or to the rest of the room, please do so in the chat box. This will go a long way in helping us organize the session during the discussion. Also, today's event is being recorded and will be available for viewing at a later time on the Foundation's YouTube page. With that said, it is time for our sessions to start, and I will hand the floor over to Dr. Winslow.
Thank you, Chad. Welcome everyone today to our 3rd webinar. We've had a successful 2 previous webinars. If you missed those, they are on FFB's website, multiple social media through Twitter, LinkedIn, those tracking through Facebook or FFB's social media. We should have multiple places that you can find those slides, videos. Please reach out if you are not able to either Odylia or FFB. Just a brief intro to Odylia Therapeutics for those of you who don't know us. I'm the CSO at Odylia. We're a nonprofit biotech focused on developing gene therapies for rare disease. So if you have any interest in learning more, please reach out. We do have a website and an email list you can sign up to learn more. With that, I am going to introduce our first speaker. While I am reading her bio, we do have a poll question for the audience that if you can go ahead and post that, Layla. Hopefully everyone can see that on their screen now. And please feel free to participate. What is your role in drug development? So while those answers are coming through, I'll go ahead and introduce Leslie Lemke-Boutcher. So Leslie is a drug development leader at Covance. She's a board certified toxicologist with over 20 years of experience developing ocular drugs. She began her career in the pharma sector, supporting projects from the first target identification through candidate selection, IND enabling studies, NDAs and BLA submissions, as well as global regulatory submission defense. In her role at Covance, Leslie leads a group of professional drug developers who, like her, bring the benefit of their comprehensive experience to clients who are looking for scientific and strategic guidance to optimize their non-clinical and translational programs. Before I turn it over to Leslie, we can go ahead and share the results of the poll so we have an understanding of our participant audience today. And majority are academic-based researchers with a number of patient groups and biotechs in the audience. We have some non-patient group funding groups and a few from the other category. All right, so with that, I'm gonna go ahead and turn it over to Leslie, who's gonna talk about her experience at Covance and the opportunities to strategically partner with contract research organizations. Leslie, if you wanna go ahead and start us off and maybe introduce yourself from where you sit in the world today.
Thank you. Thank you for the introduction, Ashley. I am actually located outside of Indianapolis, Indiana, which is where one of the several Covance facilities are located. I've spent my career kind of bouncing back and forth across the country, many years in Texas at Alcom Laboratories doing the ophthalmic work that was mentioned in my bio, and now about 5 years at Covance supporting client groups that are very similar to what we're here to talk to today. So I'm very much looking forward to the panel discussion later and seeing what we can do to help bring forward new medicines in this area. Do you have the slides up already? So next slide. What I'm really here to talk about today is some of the best practices and ideal ways to set your program up for success from the work that you do at the early bench all the way through to that collaboration with the CRO. And it's one of those transition points that many people don't automatically quite predict, in that much of the work that needs to be done to support a new regulatory filing has to be done under circumstances that just can't practically be done in a typical academic environment. So that's where the CRO world comes into play. And a prevailing message you'll see throughout my talk and those that follow are really that it's never too early to start reaching out to your CRO and talking about what your program is, what your goals are, to make sure that you can collaborate to get the best plan to go forward. So we all know that starting to think about a new drug development plan, we spend a lot of time thinking about our target and our unmet medical need that we want to address, and certainly understanding the modality that you're going to use to take forward to address that. And today we're going to be emphasizing gene therapies as our prototype example. But a lot of times there isn't quite as much understanding about the logistics and the details that need to be worked out for the regulatory environment. And this does get into quite a bit of detail about the specific vector that you're using, and having not just a answer of, you know, this is what someone used before, but a real deep science-based answer about why that vector is right for your particular indication. And then also knowing that you have a good amount of drug supply that is well characterized to meet the environmental or the regulatory requirements for each environment that you're working in, whether that is early discovery, GLP, IND enabling work, or on into the clinic. And especially with gene therapies, it's really important to understand that clinical material requirement early on, because you're making relatively small batches of expensive material and you want to be able to get the most utility out of that possible. The next thing that is in the logical order of progression of things that get considered is your species selection for your non-clinical program. And this really is a factor even in the early discovery work. In many areas of science anymore, it's very common to go into mouse models, and particularly transgenic models are a great model for disease for many indications and therapeutic areas. Unfortunately for ophthalmology, they're not quite as ideal of a model based on some academic or anatomical differences between rodents and the larger order species. So I just have this easy graphic here to see the amount of proportional space that the lens occupies in a mouse or a rat in comparison to humans or other species is quite different. And that creates a challenge for the surgical models where you're trying to introduce the material into the vitreous or subretinal spaces. In an academic environment where you're focused on looking for efficacy only, it's pretty common to not look at the rest of the tissues in the eye to make sure that you don't have artifacts of your surgical model. But when you go forward into your non-clinical program to support your IND or first in human, you will have to evaluate that full extent of tissue. And if you see things like physical harm to the lens or cataract formation, which can often happen subsequent to a lens injury, you'll have to explain those and be able to convince regulators that that is an artifact and not really an impact of your drug. So these are the kinds of knowledge and experience that you expect to get from a good CRO that's accustomed to working in the specialized area and really able to give you advice on how to best plan your program going forward. There's also some unique questions that come up around the acceptability of unilateral versus bilateral dosing. We all know that bilateral is typically the approach that we're hoping to achieve in the clinic, but you may have to, in your animal studies, do a more staggered approach in separation between the two eyes being dosed because of concerns with that early material and not knowing what kind of tolerability you might have, and if you might even temporarily cause decrease in visual acuity in those animals enough that would prevent them from carrying out normal feeding behaviors. For example, your eye cook will weigh in on that and will put some restrictions on you. And then the last area for kind of touching on in the generalities is that according to the global regulatory expectations, when you're developing most types of therapeutic modalities, you are expected to do both a rodent and a non-rodent species. Gene therapies tend to be a little bit different in that based on the therapeutic and its targeting, you may be able to justify one species. And typically what happens here ends up being that you justify non-human primate as the closest to the human environment. But again, this is something that is very dependent on your specific target and vector, et cetera. So these are detailed discussions that you should be having within your team as you develop a plan, and definitely when you start speaking to CROs about helping you pull these things together. One of the reasons that I say it's never too early to start talking to your CRO is that there are a lot of constraints that you'll be working with that are new to people that are transitioning over from the academic environment. Most of the time when we're working in a university laboratory, we're just thinking about traditional animal ordering and acclimation schedules. So as long as you get your order in time to get the animals shipped from the vendor, you can proceed with scheduling your studies pretty readily. That doesn't happen quite that way when you're looking at a CRO environment. It's kind of like trying to book a hotel room at a holiday in a very popular destination. The better the destination is, or the better your CRO is, the longer wait time or lead time you might need in order to be able to book that work. So we need to keep all those things in mind, especially because it's not uncommon to get into a situation where a new startup company will come to a CRO and say, we have a commitment with our VC that we'll have IND enabling data by X date. And they just weren't aware of some of the steps or hurdles that needed to be overcome in order to collect that data. So they find themselves in a disconnect that they've committed to something that isn't actually possible. So again, the sooner you reach out to your CRO and start talking to them about feasibility and timelines, the better they'll be able to help you with your budgeting and planning timelines to be able to make sure all those pieces come together. And of course, like in any other area, you're typically starting out with in vitro studies to confirm your compatibility or your physiologic effective intent, and then moving into non-GLP studies to start dose escalation before you go into GLP work. So although as scientists, we're always very excited to start the project and quickly move into the laboratory, when you're doing the work to meet regulatory goals, there are some additional timelines that can definitely come to pass. Next slide, please. So here I've kind of switched gears a little bit and put in some comments and things about what the CRO will need from you. And on the right, I have a graphic that we created to really try to highlight the fact that the strength of the collaboration between the sponsor and the CRO really becomes the machine that creates that drug product that you're looking for, whether it's an injectable or a tablet, in whatever indication it may be. You really need to have all those gears working well together in order to get your product to market or to the next regulatory stage at the time that you're planning on. Then the biggest one I have highlighted here is that transparency. And we frequently run into situations where academics who are trying to set up a startup actually find that they have just been really inundated by their IP department with warnings about protecting that asset and, you know, being very careful about disclosures. And then they come to a CRO to try to ask for a program to be designed and executed. And they're in a mindset where they feel that in the best interest of their asset, they're not able to tell us what their target is, what their exact asset modality type, et cetera, might be. And while it's understandable to be protective of your IP that is so precious, you know, every program for an IND enabling program, a clinical program, they really are very unique, and they need to be modified to fit the specific needs of your particular project. So that transparency is critical, or you could find yourself in a situation where a set of studies have been run that in the long run really don't quite fit what your need is. And of course, cost, timing, scope of work, quality, risk tolerance, and having the right resources engaged in the process are all going to be key. And as you evaluate different CROs, you'll find that some have strengths in one area or another, and it's not as common as you might think to see that you have a CRO that really has the full breadth and depth of resources to reach from the early discovery all the way through the clinical phase and be able to bring those expertises to your view when you need them. So again, as I mentioned, every program needs to be unique. They're really going to be dependent on the clinical program that you have designed. So sometimes this is a shock to sponsors when we start asking them, you know, what's your patient population, your dose route, your expected frequency, the duration that you think you need to dose in the clinic in order to see the results in your proof of concept study that you're hoping to see. And these are all questions that help us design a program so that we can meet all the regulatory requirements with the minimal number of studies to fit that goal. So again, if you don't communicate effectively about that, you could find out that you just finished a 2-week animal study, but in order to move from your SAD or single ascending dose clinical study into a phase 2 proof of concept, you need to very quickly turn around and run a 2-month study or a 3-month study. And that's just not as efficient, nor is it animal efficient. So those are all things that we like to make sure we take into consideration. Another thing that people frequently just don't understand is that ophthalmology programs in general, whether they're for gene therapy or any other type of modality, they do tend to be more expensive than a more traditional systemic program. And this is because of the number of additional evaluations that need to be included in those studies and the difficulty or specialized skill that's associated with those particular evaluations. And as I touched upon very briefly in the prior slide, you know, it's never too late or too soon to start these discussions. And it's really getting that groundwork in place frequently, getting the confidentiality agreements in place so that you can feel comfortable disclosing details about your product with us, getting master services agreements in place so that you understand the financial commitments that you're making and the schedule of payments or you know, payment terms, delays and cancellation fees, et cetera, that will all be included. The queues, as I mentioned, you know, if you want to go book a hotel room in Hawaii at the best resort possible, you probably can't do that in 2 days' notice. And the same is true with a good quality CRO. The other item I touched on again in the last slide as well is understanding those order of operations or chronology of tasks that need to go forward. Quite frequently, clients will ask us to start their pharmacokinetic assay development, or in the case of gene therapies, their biodistribution, method development, and validation in parallel with the conduct of the animal study. And if you are a very risk tolerant organization, that might be something you want to pursue, but your CRO should be explaining to you the risks of that approach. And, albeit something that doesn't happen every time, there is a big impact if you get done with your study and you've been collecting and storing blood samples all along, and you find out at the end of that study that there was some critical stability issue with those samples that normally would have been found during the validation, but because you chose to be more aggressive, you didn't wait to know that, and now your samples aren't actually valuable. So these are all types of questions and thought processes that your CRO should be able to help you with, and really be able to help you maximize the program speed, so that, again, you don't have to go back and spend more money and time than you could if you're pre-planning well. Next slide. This is just a brief example of what a Gantt chart could look like for a program. Again, as I mentioned earlier, we typically see programs only going into the non-human primate, but depending on your particular asset and target, you may need the 4-week rat study as well as, say, a 4-week primate study. And then most of the time for IND opening, we see 13-week studies or 3-month studies being sufficient. But again, it can take several months to get the preliminary work done before you're really in the best position to start these studies. And your CRO should also be asking you about when your regulatory target is, which here I have as the STAR for your IND or clinical trial application. And they should be trying to work with you to work backwards from that target date to when you should actually start each of these pieces, and what particular jurisdiction you're going to be filing in, and what jurisdiction specific requirements there might be that you need to take into consideration. And then, of course, in an optimal world, very quickly after you get approval from the agency for your IND, you should be set up to quickly start your clinical trial. And this is, again, something that really gives you a better feeling or visual for why we talk about it being so important to work with an organization that has in-house skills and capabilities in each of these areas. Because if you think about it, and you're not taking an integrated approach like a programmatic approach, it can create a lot of dead space in your schedule if you have to stop and re-engage another service provider for each incremental step of the program. And that's what we typically refer to as a transactional approach. In those cases, there's also an assumption on the basis of the CRO that you know exactly what studies you need and in what order they need to be conducted. So they typically won't offer you as much of the type of advice that I'm discussing today, where, again, if you take a holistic or programmatic approach, like I have laid out here, then the organization should intuitively know that it's a bigger process and that there's opportunity to have discussions with you about how to optimize the progress through this type of a scheduling system. So next slide. So now that we know a little bit about the number of studies that need to be conducted to support, in this case, the example of a gene therapy, it's very typical for clients or investigators to ask us, how do we know this and where do we get this information from? So I've included here for reference just 3 examples of overarching regulatory agencies and their guidance documents, or where to find their guidance documents to help drive this discussion. The US FDA and, of course, the European Medicines Agency are 2 very strong examples. And then I've also included the International Conference on Harmonization. This is an organization that started in the early 90s to try to get us to the point where each country on the globe would stop asking for different sets of studies or different types of studies to support a single application. So ICH has worked very hard since then to get a harmonized set of guidance documents and requirements that are accepted by all regions. There are still some places where there's specific variations if you're in particular taking your product and trying to file it in the US as well as say China and Japan. But ICH and especially the ICH M3 guidance are really key starting points for most preclinical planning. And then there are additional kind of levels of specialization in the guidance documents as well, depending on your therapeutic area and your modality. And I've included a couple examples here of just how that hierarchy progresses from the generalized guidance that's the M3 through a preclinical for all cell and gene therapies, and then how that might evolve to give a little bit of different specialized advice for clinical trials. And then as the agencies see more and more products being developed in a certain specialized area, they will also come out with even more detailed guidance for that occasion. And that's what we see here with the evolution of the FDA's guidance document for gene therapy for retinal disorders. So reading and understanding and having experience with conducting programs under all these guidance documents is a key expectation you should have for a CRO that you're considering engaging in your program. Next slide, please. The other thing that I like to try to make sure that all of the staff on our team is aware of and that we have a good understanding of with the client is, again, the regulatory environment that they work in as far as the 4 regions that we show here on the right, whether or not their organization has a variety of different modalities they're working with now or expects to evolve to in the future. And then we need to be up to date and engaged in all the different types of modalities that you might see for ophthalmology, because we learn and experience different things based on each of those modality types. And the totality of that learning really helps us understand the eye in general and overall much better than if you didn't have an engagement in each of those areas. And that totality of knowledge, again, really puts us in a better situation to be able to interpret results and the translatability of any findings that we might have to be able to help guide, is this a driver for a no-go decision for your program, or is this something that we've seen in other areas and we know that it does or doesn't translate to humans, or how it might resolve. So it's that holistic knowledge that is also key to a successful program. Next slide. So whether you're talking about Alzheimer's disease with a small molecule or gene therapy for a retinal disorder, there's some universal program objectives that you expect to get out of your IND enabling program. And these are really key drivers for being able to help you transition into the clinic, whether it's developing or identifying the active dose levels, looking at how those dose levels might need to be adjusted to move into the clinic, understanding the feasibility and reasonability of the proposed clinical route. I remember when a lot of the IVT drugs for AMD started, and we were all just shocked that anybody would consider a monthly intravitreal dosing. But certainly if you have a strong enough performance of your drug, that becomes accepted by patients much easier than we had initially expected. But because there is risk associated with many different routes of administration, all of that has to be explored and understood. And again, identifying the direct toxicities of your product and looking for potential biomarkers that might help you monitor either efficacy or safety as you transition from the animal models into the clinic are important factors. Next slide. So all gene therapy programs will have some common elements, and these are also common with the general types of non-clinical studies that would need to be conducted for any type of modality. And in a high-level sense, they're very similar to what you might experience yourself if you went in for your annual physical at the doctor's office, as far as looking at body weight, your blood work, just visual exams of your clinical presentation. Also included are monitoring of blood levels of the particular therapeutic or transgene product in this case. And again, understanding how those may change over time. What's unique when you're talking about a gene therapy program is there's an entire new class of testing that needs to be incorporated into these studies. And that is looking for what we refer to as biodistribution and viral shedding. And these are really specific types of studies that are done to understand the movement of your genetic material or product of the transcription of that material from the local site of application to the rest of the body. So where does it go to? How long does it last? What type of mechanisms are used to clear the vector as well as the transgene product? And does your body have an autoimmune response or develop anti-drug antibodies to either the capsid that the virus is packaged in or the transgene product itself? So again, if you ask somebody who is developing oral small molecule drugs how much an IND package costs, they're going to give you a certain number. Once you tell them that it's a gene therapy, the cost goes up because there are these additional evaluations that have to be done in order to really understand the safety of your product. Next slide. So again, here, when you go from a systemic gene therapy for lack of illustration, I guess, and go into ocular programs, there's going to be another level of extra tests that need to be included. And these are very similar, again, to what you might see when you go to your ophthalmologist appointment, looking at your intraocular pressure, doing a slit lamp evaluation or indirect evaluation where it's a microscopic look at the health of the front and back of the eye, electroretinograms to look at the conductivity of the visual processing pathways. OCT and histology are kind of an in-life surrogate. And then, of course, the actual tissue processing to look at the health of the tissues in different areas of the eye. So again, adding another tier of types of studies. And then when you get the results from this study, your study director at the CRO will provide you a report that is very factual based about what was seen in the study. They are not in a situation to be able to take that information and put it in context of the clinical plan or the clinical significance, because that's outside of their purview. If the sponsor isn't actually in a situation where they can do that translational interpretation, there's a multitude of skilled consultants that are independent, or you can work with an organization such as mine within Covance, where we have the in-house expertise based on working in the drug development arena and writing those INDs, et cetera, to be able to help you put that data into context and really understand what types of questions the agencies might have and how you may proactively do studies to potentially even fill some of those gaps. And then again, gene therapy for the eye, in addition to having all the standard tissues of the body that need to be looked at for the biodistribution and shedding. Now you add that you need to take additional tissues from the eye, and whether this is fluids like vitreous or aqueous humor, also take sections of the ocular envelope, which you see in this bottom graphic, splits out and shows that there's multiple different layers of cell and tissue types within that ocular envelope. And you will see that these are all dissected apart from each other and evaluated singularly to really give you a good idea of whether or not your gene therapy got to the target tissue of interest or got someplace where perhaps you didn't want it to. But is it in the right place? Is it expressing and maintaining as it should be in that area? So again, a lot of layers of complexity as you get into the more specialized type of testing. Next slide. So just a couple quick remaining slides to give some points to consider. There are indeed other types of non-clinical testing that are necessary to support IND or CTA filings. Globally, these are not typically required for a gene therapy, just based on how the body is known to interact with DNA moieties. But if you were developing an oligonucleotide therapy or a small molecular, you would potentially have to do standalone drug metabolism, safety pharmacology, and genotoxicity testing batteries before you filed your IND. And for any different type of therapeutic area and modality, you would also need to look at the potential risk for pregnant women and developing fetuses being exposed to your drug. So that's the DART, or developmental and reproductive toxicity battery, and also potentially carcinogenicity testing. These are all things that would happen after your IND and before your NDA or BLA. So again, working with a good consultant or in-house expert at your CRO, they should be able to help guide you here. Next slide. Some final thoughts before I pass off to Michelle and Tom is to really understand that if you make changes in your drug product or your manufacturing process after you start your studies, whether it's the animal studies or the clinical studies, you have to really keep a close eye on those to determine if they could have any impact on safety or comparability of your data from one phase to another. So it is possible that you may have to go backwards and do additional studies to make sure that you resolve any gaps that are there. So key point to keep in mind. And then another item that we find most people aren't aware of is the fact that you can actually go out and learn from the experience of predecessor programs. So through the Freedom of Information Act, you can go out to the FDA or the EMEA and other government agencies' websites and look for their documentation of the basis for how they made the decision to approve that product. For a biologic or a gene therapy, it's referred to as a summary basis of regulatory action. For a small molecule, it's referred to as a summary basis of approval. But I've included the link here for the document for Luxturna, just so that you can get a flavor for the types and extents of studies that regulatory agencies are prone to ask for. And then I believe I have one more slide. This one is really just intended to give some tools for after the webinar, where you can look at information ranging from relatively high-level overviews of what gene therapies are and how they work, all the way up to very detailed case studies or white papers about issues that have come up and how they've been resolved with the development of gene therapies in either rare and pediatric diseases or, of course, retinal diseases. And with that, I'll be happy to answer questions. Thank you.
Thank you, Leslie. Real quick, while we're transitioning to a series of questions, please feel free, by the way, to ask questions directly to Leslie during this section in the Q&A button that's at the bottom, kind of right-hand side of the toolbar. So while we populate questions, in the meantime, we're going to run one poll. you want to go ahead and post that one, hopefully you guys are seeing that in front of you now. And that would be, have you worked with a CRO before, just to get an understanding of the experience of the current audience. All right, while that's populating, Leslie, thank you very much for the presentation. It's extremely helpful. And I think very apt for this audience. Hopefully your experience and the relevance to gene therapy and ocular will resonate with a lot of the folks on the phone. We will have the slides up. Hopefully, we haven't posted the slides yet from the previous recordings, but we have talked about it. Right now, the recordings are up for webinar 1 and 2. I'm going to go ahead and post that link in the chat bar later, maybe once we start with the second speakers. But it's something that we've talked about in the past, is making those resources available as well. So we will check back with us, and hopefully at webinar number 4, we can point you guys to that resource. All right, so have you worked with a CRO before? Actually, 56% of our audience has, which is a significant amount of experience, so wonderful. All right, with that, I'm going to go ahead and switch over to a few questions for you, Leslie. And reminder, go ahead and submit those questions now. We will also have a full panel session later on with everyone. So if you're an academic who's never done drug development formally, you're generally more on the research side, and you want to engage a CRO, where do you start? What does that process look like? You know, does a university establish that relationship? Do you reach out directly? How have you seen that worked in the past?
Great question. So every CRO is going to be a little bit different. I think nowadays you can definitely do some online searching and see a variety of service providers' websites that you can reach out through. Covance ourselves, we have even specialized salespeople who work with innovation centers or universities directly to make sure that we do have kind of broad ranging confidentiality agreements and master services agreements in place. So kind of a preferred provider relationship to a degree, but a lot more organizations nowadays are setting those things up to try to make the process easier for their academicians.
Great. All right, question from the audience. Is the timeline you showed after efficacy studies?
So the Gantt chart that I showed, if that's what you're referring to, is typically that clinical phase would be your first in human. And the length of time that might take depends very much on the rarity of the disease that you're treating and how difficult it might be in order to identify those patients. Because of the connection between Covance and LabCorp, we actually have access to a lot of masked patient data to understand what types of testing is being requested by their physicians and where those patients and physicians might reside. We can't see names or contact patients directly, but there's ways of trying to proactively, again, understand the environment and plan your study to be able to make sure that you have the right patient population. And I see here that the person clarified they meant preclinical efficacy study, which again is a great question. It's an interesting point. It's very common to presume that you must have preclinical efficacy data in order to go into your IND, but that's actually not the case. You have to have at least a well-written paper rationale for why you believe you're targeting something of use. And the challenge and the reason that there's flexibility there is that there isn't a really good efficacy model for every disease and every modality. So much of the work that you see being done for preclinical efficacy is more being done to help the organization decide if they have the right candidate to take forward, not that it's required by the regulatory agencies. So the timeline that I provided is really just focused on the work that is required in order to have adequate safety data, and that covers toxicology, drug metabolism, safety pharmacology, that suite of studies that knowledge is required to open your IND and be able to go into your first in human patient. So any timeline that would be associated with candidate selection and optimization type efficacy studies occurring in the academic facility would be a predecessor to what I shared.
Okay, great. All right, with that, Leslie, we're going to go ahead and move on to the next section. We'll bring you back for the full panel discussion. Thank you.
Thank you.
All right, I'm going to go ahead and introduce our next two speakers who are going to tag team, I think, on this section. They're both from Aldevron. So with that, I'm going to go ahead and start with Michelle Berg, who's president of GMP Nucleic Acids at Aldevron. Michelle brings over 20 years of experience in the biotech sector. In her current position, she oversees Aldevron's strategy to provide GMP plasmids and mRNA for gene editing, gene therapy, and cell therapy applications. She works closely with the company's operational team to meet the requirements of these growing fields while supporting the clinical and commercial efforts of Aldevron's clients. Just an interesting fact and something you may or may not know about Aldevron, it's actually the GMP plasmid facility located in Fargo is the largest in the world. She is also a contributing author and speaker on patient-focused programming, rare disease advocacy, and accessible education on genetic medicines. And then Thomas Foti is the president of the protein business unit at Aldevron and is one of the original founders of Aldevron's protein service business. Over a period of 10 years prior to working for Aldevron, he served in several roles in Merck's bioscience division, most recently as the director of its global custom services business. And with that, I'm going to go ahead and hand it over to Michelle and Thomas. If you guys want to let us know where in the world you are calling in from today, that'd be great.
Maybe go ahead, Michelle, to introduce yourself.
Yeah, hi everyone. So I am actually calling in from Fargo, North Dakota, from our headquarters. So great to be coming from you live from here. Tom?
Hi everyone, my name is Tom Foti and I'm based out of Madison, Wisconsin, where Aldevron's protein business unit is headquartered out of. So also excited to talk with you today. And I think with that, we can transition to the slides. Thank you, Chad. I just wanted to say thank you to the FBB organization and Odylia for inviting us today. It's a pleasure to be working in this amazing field of genetic medicine. And it's great to serve alongside Michelle as we try to build our business and help support different organizations make the breakthroughs that they're working on. So if you can move to the next slide, that'd be great. Just to give you some context of Aldevron as it relates to this topic, I think it helps to give a little background on our company. Aldevron has really been a pioneer for a long time, manufacturing different types of biological materials, mostly focused on plasmid DNA, and then in the last 10 years, focused on proteins and messenger RNA. And so we've been around since 1998, and through that process, we've learned so much. And successful projects, certainly many of them, the things that didn't always work out correctly, working with different clients. And we are happy to share some of those ideas and thoughts that we've learned about through the years in today's program. We have over 550 people who work for Aldevron based out of Fargo and in Madison. And together we're working super hard every day to deliver on the mission of making a big difference in this world with genetic medicine. If you move to the next slide, just a comment about our vision, and I want to tie it in directly to the idea of working with a contract development and manufacturing organization, and I'll be hitting on those topics as we move through the slides here. But what we are trying to do is we're trying to enable our partners who are our clients to do amazing work. And our tagline is the basis for breakthroughs, which basically speaks to the idea that we're manufacturing these materials that are going into these different types of genetic medicines. And together we partner with these clients. And this partnership is super important because we bring a certain level of experience to the table, but so do our clients. And so those of you in the audience today that are thinking about what a contract development and manufacturing organization brings to the table, it's just not a facility with hardware that can deliver product at the end. It's really a relationship where over time together you learn from each other, and I think together you kind of partner up to do amazing things. So we really seek to be a partner of choice, and if you're looking for a CDMO to partner with, that's an important thing to be thinking about. Can you move to the next slide, please? And so I talked a little bit about our products and services and how they enable this new world of gene therapy and cell therapy. And on the left here, you can see all the different therapeutic areas that we're working on. You know, many of these are applied to ocular diseases. And Leslie had a slide there with kind of the breakdown by different therapeutic approaches. And so on the left, we talk about cell therapy, gene therapy, messenger RNA therapy, which is an up and coming approach that many of you are aware of, I'm sure. Gene editing therapy around CRISPR-Cas9 and guide RNAs and the ability to deliver RNPs to engineer new cells and cell therapies. And then vaccines is the other area that we're focused on. And so those are the things that our clients do. But in the middle, we kind of aligned some of the products that we deliver into those markets. And so we think about how we need to deliver those, not just at a research grade level and a preclinical level, but also as our clients experience success. One of the things about Aldevron is we can progress through the different stages of drug development with the client, supplying material at research grade, a GMP source level for different early phases of clinical trials, and then also manufacture commercial product for in full GMP suites. So that gives a little bit about our background. If you want to move to the next slide. And so the main topic today is to talk about elements of a contract development and manufacturing organization. And Leslie was talking, you know, about CROs, which are contract research organizations. Sometimes you see the acronym CMO, or contract manufacturing organization. And whether it's a CRO, CDMO, or CMO, one of the most important things I wanted to re-emphasize, and Leslie brought this up also, is this idea of this long-term thinking, meaning that you don't move directly into getting filing an IND or a BLA. You have to do a lot of work before that. But it's very important early on in your research program to think about what the end goal is. And there are many reasons for that. And so different types of contract development organizations may have different levels of experience in making sure that what you're doing at the research grade level is going to be eventually GMP compliant from a manufacturing standpoint. Another thing you really want to think about when you are talking about a contract development organization is this idea of many times most of our experience is in contract biological manufacturing, not specifically contract small molecule manufacturing. And one of the things with the progression of large molecules with antibodies being a major component of pharmaceutical sales nowadays, and now moving this new world of genetic medicine, we are trying to, and the world is trying to manufacture different products that are in biological systems. And we're working on this concept of trying to engineer biological steps so that they're very consistent every time. And so when you're talking to a CDMO, you want to be thinking about if they're thinking with that type of mentality is exactly the types of conversations that you're going to want to have. Capacity is an important topic to have a conversation about, is because it's amazing now with the different types of genetic medicines where in some particular cases, and this is very relevant in the ocular market segment, is that sometimes there are diseases that are relatively rare, and sometimes there are diseases that reach a wide population of patients. And depending on, again, what the end goal in mind and maybe what the size of the market, you should be thinking early on around the capacity and the potential batch sizes of materials that you need to be made to be able to deliver on your different phases of your clinical trials. And so there are situations where for a phase 1 clinical trial with maybe 10 or 20 patients that might be enrolled, that the client needs relatively very small amount of material. And sometimes large CDMOs might say that your requirements are so much smaller than our smaller batch sizes that we're not a good fit for you. And so that's going to be a conversation that you're going to want to have early on in the game. I wanted to talk a little bit about QC tests, because the assays that may work in a research grade setting, in a GMP setting, they need to be validated in a way so that they are consistently executed and reliable for product release. And so that's a lot of times people think about the manufacturing aspect also, but it's very important to also be thinking about the QC tests and assays for product release. Here, the last bullet talks about manufacturing and QC information that is required for regulatory filings. And so you should be asking your CDMO partner, if we are successful with on the science side with delivering, safety and efficacy, if when we do our filings, what access will we have to information that will be required to fill out our CMC sections for our INDs or additional regulatory filings, maybe in different regions of the world, where the manufacturing organization needs to supply this information for our client or the sponsor to be able to do the work that they need to do. And so having a good conversation about that on the front end is very important. If you can move to the next slide, I appreciate it. So the next slide, how to identify a CDMO. And I can imagine that this could be a daunting task, especially for maybe companies that are relatively small in early stages of biotech startup world. Or we also have worked with, and it's an evolving market for us, are well-funded translational medicine organizations that are within a university setting. And so here are some things maybe to think about when you're identifying a CDMO. The thing about when you work with somebody to do these very special projects that you're totally invested in is like figure out who you can trust. And it's kind of a little bit of the old age game, is that strong referrals are super important. And we say that because we have a lot of experience on the Aldevron side. Is basically that's how we built the business. We have invested very little in sales and marketing relatively to our size over the last 20 plus years. And we spent a lot of money on our manufacturing expertise, our quality expertise, our project management and client relations. And because of that, we've gotten a lot of referrals. And so I would suggest that you spend a fair amount of time just going out to your network and finding out who is a good company to work with. And if you're in Boston and San Francisco and Seattle and San Diego, Philadelphia area, North Carolina, Houston, where a lot of the biotech is going on in the world, there's often conversations that you can have in user group networks around those areas about who might be a good CDMO to work with. Of course, there are conferences like bioprocessing conferences that focus on different companies that are in the business of manufacturing. And I would suggest that those are very helpful to go to. There are contract manufacturing journals that are out there that are very helpful. And so those are just a couple of ideas on what you want to, on how you want to address the search basically for a CDMO. And once you find somebody, so now you're into the how do you engage with them. And often these things start with a CDNA or some type of, sometimes MTA, if there's some type of tech transfer that is going to happen. And so it's important from the beginning to get the right legal structure organized to make the business run smoothly and the science exchange to be collaborative. I would encourage the idea of using a pilot project early in the process, so that once you get these agreements in place and you get a good work plan together, is to do a small scale project. And that is one of the reasons we have a research grade area, and we can do projects that are cost effective and relatively quick, where we can turn around this a project in weeks, not months, to see if there is a kind of proof of concept that the process works for the intended purpose. And so pilot projects are super helpful. People use supplier surveys to engage, and those are important, especially as you're building out your, let's call it your confidence in the quality system of a CDMO, and conversations of getting quality people involved on each side of the relationship is important early on. We also offer, as a company, and most companies now do also offer virtual audits. Virtual audits give you a flavor of the facility in today's world of COVID, when people can't travel. These projects must go on, and so we are working on mastering the kind of the delivery of virtual audits for clients. And the last thing I want to talk about here is the opportunities and benefits of working with contract development and organization. And there's a couple of obvious ones, especially if you're smaller. You may not have the infrastructure from a biomanufacturing standpoint to be able to deliver on the production of materials that you might need for your clinical trials, or your proof of concept trials, preclinical. And so, so obviously partnering with somebody who has that in place is important. That can save time, it can save money. And another thing that people do with contract development organizations, and of course they do this with Aldevron, is they leverage our network. And so when you've been in business for 20 plus years, you develop a network of all different types of suppliers and vendors. And we do a lot of QC testing in-house, but we don't do all of our QC testing in-house. So we have a network of vendors around just quality control that are important to us. And the value is that we have qualified those people in our quality system and have audited them. So that is a big value that a CDMO can bring to the table to help maybe fast track your program. I would say those are the main things that I want to focus on this slide. And so moving to the next slide is just a timeline. It's similar to what Leslie had mentioned, that, as you move through these different stages, you want to make sure you do a couple of things. You want to understand the timeline. It's very important, because a lot of times people have unrealistic timelines on what's possible in a certain amount of time. And so that's one thing to be thinking about. You want to be thinking about scale. I mentioned that earlier. You need different scales of material here at different stages of the process. And so if you're successful, you want to be thinking, at least a year in advance, what type of scale you need to be manufacturing for your next stage in this progression from, a concept to actually a commercial FDA approved drug. And the other thing I wanted to mention is that this often is not thought about until there's a problem, is have you evaluated a secondary vendor besides your primary CDMO? And, there are changes in companies all the time with people, and sometimes key people leave, sometimes companies are bought. And it's just a good practice to be able to identify a secondary vendor for all these different types of work, and for that matter, a secondary CRO also, as you progress through your different stages. So moving to the next slide, a couple other things that just to hit on to add to my previous slide around benefits of working with partners is that you can have parallel work streams, which means that they're working on their area of expertise. You're maybe working on testing things, which is your core knowledge. And together you can parallel work plan some of these different activities so that you can get things done faster. I talked a little bit about leveraging complementary expertise. Again, it's nothing like somebody who's done this before. And, you know, Aldevron has produced over 3,000 different types of recombinant proteins. On the DNA side, we've produced over 140,000 different lots of research grade material for plasmids and over 2,500 clinical manufactured lots of plasmid DNA. And all the learnings through that process is something that is something that we, you know, can benefit future clients. Talked about the things of the benefits of working with different types of CDMOs. And, you know, at the end of the day, who we're really working for are the patients. And if you have an excellent idea and can get things done faster, you know, we're all really kind of in this business to benefit the patients with, unbelievable genetic medicine. And so those are a couple of things I wanted to focus on there. Considerations for selecting the right CDMO. I have some things listed here. You know, there are different types of regulatory requirements in different countries. And it's just something to be mindful of, that if your CDMO has experience delivering materials to global markets, this can be very helpful if you plan to do clinical trials in different parts of the world, which is often the case. We talked about reputation and referrals, and you know, asking people about, you know, how do they keep track of the client experience, and their net promoter scores is one example. You know, I like to ask the question in regards to how engaged are the employees at a particular CDMO, and some employers will do engagement surveys, and you may not get all the details of those different types of situations, but you get a sense of what a great partner might look like. The other thing I wanted to mention is just about cell lines and freedom to operate. And so as I talked about earlier, experience with a particular CDMO who has done this before, research grade through commercial. And as you have those successes, good conversations about the plasmid DNA, the host systems, the original synthesis of the gene, how that happens, and thinking about if you progress to different phases as you have success with your program, does the CDMO or does the sponsor have the freedom to operate? Do you use those materials to manufacture the products that they're interested in sourcing? So something important to be thinking about. The last slide before we pass it over to Michelle is just a slide that kind of parallels this idea of scaling from smaller scales where you're doing proof of concept research grade experiments. And as you progress, one of the things that Aldevron has pioneered is this concept of this GMP source quality grade. And that's in the green here, it's highlighted. And as clients continue to have success with their clinical trials, we then move to offering a full CGMP offering, which clients will for sure need when they move to commercial. And the different sizes of these fermenters, and from shake flasks to fermenters, is just the thinking around, as you scale, make sure that the person who you're partnering with can scale along with you. And so with that, I will stop and transfer it over to Michelle.
Right. Thanks, Tom. Thanks so much. And again, I just want to express my gratitude to FFB for putting this together, Odylia, as well as Regeneron for sponsoring this series. This is a topic that is something I'm quite passionate about. And what I mean by that is sharing about manufacturing, the steps within, because I think that's somewhat of a black box to certainly to patient organizations that I've engaged with, and potentially as well for those that are emerging technology out of academic research labs, or as you're just getting started and launched as a brand new biotech. And so I'm really excited to continue the conversation here. And what I'm going to share more about is how you prepare to engage and work with the CDMO, important considerations to make sure that there's success, not only in reaching that first goal, which is getting into the clinic, but thinking well beyond that, as Tom and Leslie both shared, that the considerations and the things that you can do early on that really pave a way for commercial success. And then we'll share some real brief case studies, because that always gets people's minds going. So if we can go to the next slide, Chad, that'd be excellent. Thank you. So I won't restate everything that Leslie shared, but I want to reinforce that thinking about the patient population, thinking about how your potential candidate could be delivered is absolutely critical from the very beginning. And I'd like to say that this is a great opportunity to engage with a reputable patient advocacy organization like FFB, for example, to really understand the patient experience, progression of the disease, severity of the disease, and really that helps you to define the mechanism for delivery, eligibility criteria, as Leslie shared, but also to Tom's point previously with regard to scale, it helps you to understand the patient population, incidence, prevalence, such that you'll have an idea, to kind of restate what Leslie said, keeping the end in mind, you'll have an idea of how much from a scale perspective will be needed, and can therefore work backwards in designing your CMC plan. And so understanding these items at the very beginning is truly going to help you with success all along the way to reach, even the potential for commercial. So with that, I'll have you transition to the next slide, please. So I'm going to be coming at the perspective of design for the plasmid DNA as you're considering how you're putting your candidate together. And it does help to engage with a CDMO as early as possible, as was shared, because a reputable and an experienced CDMO will be able to help you through and navigate some of these different questions as far as the design aspect goes. And so when you're thinking of your gene of interest, oftentimes there's limitations with regard to the size that can actually be put into some of these viral vector delivery mechanisms. And I refer to adeno-associated virus as one of the most common that's being utilized right now. So really thinking about the gene that you're interested in and the size and other elements within is going to be an important consideration as early as possible. To make sure that you're actually delivering what you want to deliver and where. The origin of replication, I would also add into that the promoter that's selected. That's going to help you dial into not only success with regard to yield, but also the level of expression. And so depending on where you're headed, if it's the eye versus, somewhere maybe perhaps systemically, those are considerations that you'll want to have in mind as early as possible when you're in that design phase. Because the theme of this is that the later that you make changes, oftentimes what you see, the more costly or more expensive from a time perspective, and therefore reaching your patient population that becomes. Additionally, we do still see that the agency, although it's not an absolute, does favor a non-beta-lactam antibiotic selection marker. And so if you have that option and you take that choice early on, that will be, again, another element that secures a smoother path. And that next item, sequence verification, assuring yourself that you know what you have and that you prove that that's what you have, again, is something that's going to be quite critical. And anytime that you can do standardization and prepare, as we stated, for things early on, such that you can continue forward with that standard approach is going to be better. We do see and hear oftentimes from the regulatory agencies that CMC for genetic medicines is absolutely moving faster, and therefore you must think about that early on so that you aren't making those changes later. That's something that we hear quite often. And then lastly, I always like to mention thinking about what you're calling your constructs, thinking about how you're naming them is really important, because that does tend to stick. And something as little as an I or an L or the number 1 can cause errors if you're not careful. So really thinking about how you're having a very clear name and how that is incorporated in is another consideration. So I'll have you switch to the next. Thank you. So on the other side of that, okay, so now we have what we need to consider for successful release, or what the criteria would be for the material coming out of the other side. And so that's where we really look at these items called critical quality attributes. And these are things that are going to contribute to the performance in manufacture, the safety for, of course, the patient, which is of the utmost, and then looking at what residuals come from whatever process is being utilized to manufacture. So understanding those profiles and setting your specifications accordingly are going to be very critical once again. And again, considering that you are right sizing, to Tom's point, with regard to the phase of study and marrying that, aligning that with regard to the quality manufacture. Certainly at the preclinical animal study phases of things, you don't need to have that full quality assurance, absolute review of every document like you do for GMP manufacture that will go into a patient population, to humans. And so having that right size, and then the scale and quality alongside of that is going to be very, very helpful. Tom mentioned the qualification and validation of assays as well as processes. The other thing to think about is the necessity for material to complete long-term stability studies. And that's something that I do want to urge, because I think that oftentimes does get overlooked. And certainly looking at the necessary documentation and information that is going to be needed in support of IND and BLA filings. As Tom mentioned, I want to underscore that the ability for an organization, a CDMO, to provide that, as well as the experience of their team in engaging and supporting regulator questions, IND submissions, et cetera, is going to be absolutely critical to the success, and can actually be an accelerator for clients that are seeking to drive their investigational new drug forward. And so that we can go to the next slide. So I will not go into significant detail. I want to keep an eye on time here, but much like good fences make for good neighbors, really good, strong agreements make for really good, strong relationships. And so exploring with your CDMO partner the need for, of course, a confidentiality agreement. In some cases, your institution may require or want you to have an MTA, which is a material transfer agreement. And then, of course, master supply or service agreements, or quality and technical agreements. Again, I would urge you to engage with your institution to understand what is required, but these do help to give shape and mechanisms for interaction with the CDMO, and are important to establish as early as possible and to allow time for. And so some of the guiding principles that I wanted to take you through is that ultimately the sponsor is responsible, regardless of whether you're making it yourself or if you're engaging a CDMO. And so that's something that I think oftentimes does get lost, and really want to look at it from a quality aspect, of course. We like to say quality eats cost and time for lunch. And so if you lead with quality, all else will follow. And because of what we're doing, manufacturing and biological systems, the fields of gene and cell therapy are still fairly nascent and new, that it is important to expect the unexpected, because sometimes things will go and give you a surprise. And so therefore, communication, and really strong communication, is absolutely mission critical, as is experience, to the point that Tom has made. And I want to reinforce too, something that Leslie shared, that knowledge transfer is going to be required. It's kind of that thought of help us to help you, and we're not going to be able to be successful as quickly if we don't understand where we're starting from and the lessons that you have learned. So I think that's something that's really important to express as well. And I'll have you go to the next slide, please. Thank you. So I do want to spend a little bit of time on transparent communication, timely communication, because, as I said, it's absolutely mission critical. And so when you're seeking a CDMO, I really encourage you to look for an entity that's curious about your needs specifically, someone that seeks to understand and hear what your science is, what you're aiming to do, the patient population that you're aiming to support and reach, and how. Oftentimes you find yourself in a situation where you have an individual or you have an organization that is telling you more than they're actually listening. And a really great CDMO is going to take that opportunity, again, to hear you, to understand the issue, to understand the problem or the work that you're aiming to do. And certainly, if they are engaged in the field with which you serve, all the better, because that demonstrates that there is that empathy, that there is a very strong pulse, finger on the pulse of what's taking place in the context, and that will help you to drive your program forward. And as I mentioned, there are going to be challenges. Oh, go back. Thank you. There will be challenges. So the character of an organization is really going to be telling when those challenges arise, because of course, when things are going swimmingly, it's very easy to be great. And so I encourage you to dig in and ask those questions to understand how a company or how an organization responds in the face of challenge. And certainly looking at that lower right quadrant, understanding the cost of change, as I mentioned, any changes later on do definitely take a hit with regard to cost, but as well time. And so asking those questions and really taking the time to put things forward in a manner that will offer up the best yields, offer up the best stability early on is going to pay off dividends on the back end. And an experienced CDMO will help you to do that. And then lastly, working with a group that has a service mentality, that seeks to grow and establish trust with you, that has excellence in project management to make sure that the communication is free-flowing, that there is a responsive, connected set of stakeholders on the other side, is going to be, as I said, mission critical to accelerating the development of your investigational drug. And so with that, I'm actually going to have you transition to the next slide and bring back Tom to very quickly go through the case studies.
Yeah, so I just, there's nothing kind of like an example to learn from. And so we have one here. There's an interesting dynamic that's going on in the world where actual clinical trials are being conducted more and more at universities that are well funded. And these universities are amazing institutions. They may not have all the resources that are required potentially to do an IND filing, and that's why they need to basically partner with somebody that has that expertise. At Stanford, Matthew Porteous, we first of all, we take confidentiality very, very serious at Aldevron, and so often we can't share case studies. But in this particular case, Dr. Matthew Porteous at Stanford often cites Aldevron as his partner in the different talks that he gives. And so this is why we just wanted to basically link this case study to that source. But we collaborated early on. We offered research grade material. We offered samples to make sure that in this particular case, he was doing gene editing on sickle cell. And so he was able to make sure that that worked from a proof of concept. Even at research grade, we offered things like TSC, BSC statements that basically, in a sense, are required certifications to ensure that the products that we're delivering are animal-free. We scaled up with him to do tox studies and offered material for validation runs. And this is where we offered GMP source material to do these validation runs. So they're editing live cells. They're not putting those cells back into humans, but they're going through basically a dry run in a sense to make sure that everything works well together. And then moving to GMP supply. And recently he filed an IND that was successful. He couldn't have done it without our support related to the quality documentation that we delivered. And what often happens, and as part of our business strategy, is to work with these high-end institutions, and some of them will have success, and to the extent they do, they want to get this technology to the masses. And to do that, companies are often spun out, and then they bring Aldevron along with them as they spin out. So that is just an example of the support we've supplied to in a translational medicine setting. And I'll pass it back to you, Michelle.
All right, next slide, please, and we'll keep it brief because I know we want to get to the questions that might be out there. So to the point that Tom made, confidentiality is a priority, and so I won't be disclosing this particular client. However, it is something that we see quite often where we start at the small scale in a more research environment to provide samples of each candidate to go into in vitro and in vivo testing with the goal of coming out the other side with a winner. And so once that winner has been selected, we can then take that and as I mentioned, we put the time into that second step to understand what conditions might give the optimal yield, maintain stability of the candidate, and be able to have success for the long haul. And then we take those combinations and we generate a master cell bank from which all material can be derived for the duration of the program, from early phase clinical study to commercial, should that be the ultimate outcome. And along the way, we have taken that material such that we can optimize at scale to make sure that, again, we're really seeing the yield and the stability continue to hold true. And with that, we have done in that step 5, executing on establishing the master batch records, completing the engineering runs, and validating the release criteria, and moving into GMP manufacture and scale, such that we are providing for clinical throughout the different phases of development, and then landing on, as in the previous example, IND support that is so necessary. And so this whole series of steps with the right CDMO can be completed to some extent in parallel or overlapping to some to the certain steps, and such that it's not going to be an exact linear series of events. And so that's another way that the right CDMO can help accelerate. So with that, I'll close my case study. Thank you.
Thank you both, Tom and Michelle. We're going to briefly go through a few direct questions for your section of the presentation, and then we'll switch over to a full panel discussion. Why don't we go ahead and run the last poll question, and I think that should be popping up on your screen. Have you ever worked with a CMO before, or CDMO? We can also include in that. We'll give a second for those answers to come in. All right, while we're doing that, Michelle and Tom, if you guys could go off mute or one at a time as you go to answer questions. I'll go ahead and start with a few. Can you explain how a virtual audit works in a little bit more detail? What does that look like? What's the normal process?
Why don't you go ahead, Michelle, just because there's more audits up in Fargo in general. We recently had a virtual audit in Madison also, but they're more geared toward our GMP manufacturing facility.
Yeah, so actually our QA team has pivoted really well in this time. We certainly miss welcoming folks onto campus and giving a walk through the facility and hosting the audits. But really what happens is the team works to understand what the objective of the audit is, sets up the necessary SOPs into a private SharePoint so that they can be accessed through that portal. And then also has done a really great job of capturing video of the different areas of the facility that would typically be accessed during a tour or a walkthrough. And so, that depending again on the objective, usually is about 1 to 2 days. In a virtual environment, there's opportunities taken to provide breaks for just getting up, moving around, bio breaks, et cetera. But otherwise, it really is conducted very similarly to if you were on site. And we've gotten great feedback too, which, that's been important, that the clients that have participated have really been pleased with the results, feel that the video quality and the areas that are being shown, really have resonated and landed well, despite the video or the virtual nature.
Great. And Tom, you mentioned working with qualified vendors. Can you explain what that means for those who might not be more familiar with qualification?
Sure. And so well-organized, reputable CDMOs will have a quality manual and quality systems. And one of the components of the quality system is oversight in a strategic way over their vendors, which are bringing raw materials into the manufacturing suites. And so when we talk about qualified vendors, there's a list of qualified vendors that our purchasing team must purchase from for certain types of applications and for GMP manufacturing. And so we qualify them as a series of steps that needs to get done. We have somebody in our quality assurance department that is leading that function, and we will audit those vendors and go on site. We've also now done virtual audits of them, so we're learning also from our vendor base on how to do that. But what we mean by qualified vendors is that the material that they deliver into our manufacturing processes is fit for a purpose and is acceptable to be used in our facility.
Great, thanks, Tom. And with that, I'm going to open it up to our full panel discussion. Chad and Leslie, will you join me for on video and off mute for a panel discussion?
Absolutely.
All right, and a reminder to the audience, please feel free to submit questions through the Q&A button at the bottom of your screen to the right. All right, thank you everyone for joining us. Thank you again for your really, I think the discussion, the content, the resources you pointed to has been very helpful in laying out the value of a CRO or CMO in the development process. I wanted to touch on, at a high level, kind of backing up from what each organization does, maybe each of you can comment on what the strategic value is, if it's time saved, if it's money saved, and how you've seen organizations be able to move quicker or at a higher quality standard. It'd be great to hear a little bit more from each of you on that topic.
Okay, I can start. I know with the specific portion of Covance that I'm associated with, which is referred to as early phase development solutions, we really are the programmatic and scientific strategy kind of hub for the non-clinical side. We have similar expertise on the clinical side, but within the non-clinical programs, we've done a number of studies over the years and actually have found data to support that we can get the drug into the clinic 30% faster with the programmatic approach, where you're really thinking very data delivery key milestone versus having to wait for a full report and then transfer the report of one study and those sets of findings to another organization to start thinking about that next step. So all those pieces together where we can tighten up the white space on that Gantt chart and really start from, as I said, kind of key data sets and not waiting for final reports, when you're closer to the data, you see it faster and you can make those decisions faster with the client.
Maybe Michelle, you can also go next and comment.
Yeah, thanks, Ashley. So, I think the ability to do certain activities, either, as I was saying, overlapping or in parallel, it's a very similar theme with a CDMO that you can do that. I would also say too that, there are certain limitations to DIY, I guess is how I'll put it, in that scale, capacity, and potentially even knowledgeable and experienced hands to do the work can become the bottleneck. And so working with the CDMO who can move things in parallel, but also has the capacity to move forward with multiple programs at different stages, that can be a game changer and an accelerant. And especially when you're thinking about the ultimate group that's waiting, it's certainly it's patients. And so that is something that I think is a huge advantage.
And Tom, would you like to comment?
Yeah, just one comment. These guys hit up on a bunch of key areas, but it's very disappointing if one of our clients, for whatever reason, they come to us and they are a year or so into a program and they find out that they engineered a method that is not GMP compliant. And then they have to go back and they have to do equivalency studies. And a specific example of that is when you're selling recombinant proteins, it's also very important to have an untagged recombinant protein. And often in research grade, people have tagged recombinant proteins. They have a his tag on them because they're easier to purify. But people then use that method to go to produce material that is then in a phase 1 clinical trial. And you can have nickel that can leach from a his tag column into your product. And now you have to figure out how to prove that there's no divalent cations in your eluate. So, that's just an example of why it's really important to engineer a method that is GMP compliant if that's your end game while you're in the preclinical phase.
Great, that's a really helpful example. And Chad, do you have, do you have anything to add from your experience in the programs and groups you've worked with at FFB or before your time at FFB as well, just from your point of view, the value of adding a CRO or CMO into the equation early?
Right, so I'll comment and then I have a follow-up question sort of to tag onto the back of that. And before I answer, I would also like to encourage the audience to please put your questions in the Q&A box on the Zoom tool so we can continue to drive this discussion forward. And yeah, I think that the one thing that we're seeing is we get down to the programs that we fund. So no matter if it's small molecule or gene therapy, that they're getting to a point where they've done some animals testing, they've done some safety studies and things. And now it's like, well, what is our road to IND? And a lot of times what we want to do is validate those studies in someone else's hands. And I think that what we're trying to push some of our awardees towards is starting to think about that earlier in the process, including developing a TPP very much early on. So this is why I really enjoy this webinar that we're holding here, because I think that this really lays the foundation for our awardees that they can go back and reference this knowledge to help in their plan. While many of them, are excellent academic researchers, we have to get them to start thinking more on the product development side of things. So this goes directly towards that. The one thing I want to follow up with is you've really laid out some great information about sort of what to expect from a CRO or a CDMO. But there's so many sort of choices out there. I think for some people who are just getting into this sort of and looking into working with you all, it's a little daunting for them at times. So could you maybe lay out maybe a couple of red flags people should look for before choosing to work with any particular organization? Is there, or maybe not red flags, or something that they should maybe expect to see when doing the for lack of a better term, sales pitch?
Yeah, so I'm happy to start there again. I think one of the things that many researchers aren't aware of is that an organization, as Tom and Michelle talked about, their net promoter score, that's one metric, but another key metric is their regulatory audits. So organizations that are conducting studies under GLPs or good laboratory practice, which is a requirement for non-clinical and supporting drug development packages, go through a routine schedule of evaluations by the authorities, like the audits that we were talking about earlier. And then there's also for-purpose audits as each new drug gets to approval, and they need to verify that that data really was present and as strong as they expected it to be. Believe it or not, even in today's day and age, I've had clients call me and say, I ran a set of studies at laboratory X, and when we went and filed that application and the regulatory authority audited, they found that the data was not what it was supposed to be. And the agency came back and said, you have to go repeat all this work because the documentation isn't sufficient, the quality control, all of that isn't present. So really asking the CRO for copies of their regulatory audit history, and you are within your right to ask them if there are any organizations that you've worked with that would be a reference. Again, as Tom and Michelle said, we do have to be very cognizant about confidentiality, but even at Covance, we do routinely get that question, and we'll have clients that we've worked with and have a strong enough relationship with that they are willing to have that one-on-one conversation with a peer in another company and say, here's the things that help, or here's, watch outs or things that you need to think about. So definitely make sure that they have a long history, that they can actually demonstrate that the work that they've done has been successfully used to support a regulatory approval. I think nowadays there's a lot of very small CROs starting or trying to get established that may have great intentions, but don't necessarily have that history and experience to really bring you the quality that you need. And you definitely need to be aware of that.
Michelle or Tom?
Chad, I can add to that. I would consider a flag if you are engaging with a CDMO and they, guarantee that you do this, this, and this, your IND will be allowed. I mean, that's a huge red flag. Because going back to the point that I made earlier, the sponsor is the ultimate responsible party. And while that might be daunting to use where those previously provided, it's an important aspect of the relationship. And so, those bold statements, I think would be, to me, a very big red flag.
I think a red flag, Chad, it's a good question because, when you've done this a lot, we've been more like, they've been asking us the questions, right, in a sense, over the last 20 plus years. So we know what people ask. But I would ask, if I was a sponsor looking for a CDMO, I'd ask them to meet their head of quality. We talk a lot about quality, and Michelle talked about that. He's cost some time for lunch. And even simple questions like, can you share your quality metrics with me? We don't necessarily have to send you the data or whatever, but at least maybe a slide that demonstrates it, to share it. Some companies will not give access to maybe the head of quality, prior to getting a purchase order, and that's not how Aldevron likes to work. I mean, we have a fantastic vice president of quality and regulatory. His name is Ken Bonnell, and he's a great job in front of clients. And, I would ask a lot about quality. And if they don't give you access to quality people in that functional area, I would say, hey, I want to dig a little deeper.
Thank you.
Wonderful. That's really helpful tips. What is one, I guess, service or aspect of your companies that you think is underutilized, that people don't really tap into or know to tap into enough?
So interestingly enough, being at an organization who has been around as long as Covance has, coming from a toxicology standpoint, I always knew them as toxicology before I joined the company, and then found that there's other people who know them as a clinical trial provider or a clinical lab support organization. So I think it's it's key to ask questions and really understand the breadth and depth of what a particular CRO can do. What many people don't recognize about Covance is as the industry has evolved and the primary research has started to shift from larger pharma companies into the innovation centers and that coming out of the academic labs and hubs, we really tried to keep an eye on that and understand how the shifting needs of our client base were going to evolve. So Covance has really tried to set itself up to be the virtual R&D department for a biotech startup. And we, of course, still support large pharma organizations, but everything that you might see within a large pharma organization as capability to get a drug to market is pretty much present in Covance, with the exception of manufacturing, just because there are so many different types of modalities that we work with. It makes much more sense to, leave that specialty to people like Tom and Michelle, who can really focus on a given modality.
It's interesting you say that, because it ties back to one of the points we were discussing before, and the strategic value of working with CROs and CMOs early. One of the things that, from where I sit, happens a lot. You have startups that spin out, that get bought up, that there's a lot of maneuvering in the space that can happen very quickly. And actually, sometimes there's a real advantage to having those studies done external to that entity. It makes the continuity generally a lot smoother. Things can continue under that entity while the financial maneuverings are kind of occurring on the other side. So in some ways, it really allows the field to be more nimble and flexible.
Absolutely. We have examples of clients where our staff has worked as that point of science and strategy and program management for that company longer than any of their own employees have, just because, as you said, biotechs evolve and move on very quickly. Another thing that Covance has that evolved out of this same type of rationale that you mentioned was we frequently saw large pharma companies coming to us and saying, we know about the quality and consistency of work that you produce. I'm interested in therapeutic area X. Are you working with a startup who has a good asset in that area, and can you tell me about it? Which, of course, for confidentiality reasons, we can't. But in response to that, we evolved a new tool called Covance Marketplace, which is essentially speed dating for molecules. People who are actively working with Covance can post their non-confidential information to this site as a provider. And then the large pharma organizations that we work with for, large consistency programs, excuse me, they're registered in this system as what we refer to as seekers. And the benefit, it's not that different than many other partnering sites that are out there, but it's that exclusivity to the provider, the seeker, knowing that the data coming to support this molecule came from Covance. So they have more confidence as they start that due diligence process that they're not going to find a bunch of snags or hiccups in the quality and consistency of that data package or the records associated with it. So if the two make a connection, it goes off platform. Covance doesn't charge for it, ask for a finder's fee or whatever. It just becomes an opportunity for us to ask to be able to continue providing that service as a point of continuity as that asset changes. And, we are still that institutional knowledge to help bring it. So lots of different things can be embedded in your CRO, and it's really just important to be able to have that transparent conversation about what your needs are, and they should be giving you advice, not just asking you, what do you want me to do, and carrying forward. It really needs to be much more of a strong scientific and operational collaboration.
Yeah. Leslie, that's such a great point. I mean, that just really traces back to establishing and building trust with your CRO, CDMO. And I love the reference to the biotech matchmaker. We've had opportunity to play that part as well, but not so officially. So I might have to steal that concept. That's pretty cool. likewise have seen, you look at the field of gene therapy, for example, so much is still coming out of academic and research hospitals, institutions. And I think that will continue to be the way it will be for a certain time until that de novo synthesis comes from the companies, the NUCOs themselves. But have seen something, really interesting, great idea, working with patient groups. I mean, that's how much of this has started. And then transitioning into a small startup company that has then since been picked up by a larger entity. I mean, there's so many examples of that. And to be that source of continuity, to usher that through has been really rewarding for us as well. And just thinking about the untapped resources, or maybe just to answer your question, Ashley, about what goes underutilized or maybe is just not known. I think that opportunity to build and really work and partner in the very early stages, we love that, to be able to think about, well, what is the end goal? And then how do we build something that will successfully get there as swiftly as possible in a meaningful way for the patient population that's going to be potentially served. And then the other piece, and I really want to reinforce this, I don't know that people think about stability studies and the material needed as much as they should. And it's a real hard thing to do as an afterthought, because what happens is that you have to either eat into what's necessary for clinical trial, that's heartbreaking. That's a, real, that's unfortunate, or you have to stop and generate more. And sometimes that decision isn't made until it's almost too late. And it ends up being costly from a time perspective as well as financial.
And just to add on to Michelle, Aldevron has a service we call it storage and stability. And so Michelle talked about the stability testing for ongoing post-manufacturing. And a lot of times when people think about CDMOs, develop a process, manufacture, but like the storage of that material post-manufacturing is super important. And we have a GMP compliant storage facility attached to our facility up in Fargo. And so anyways, that can be underutilized. It's just not the stability, it's the storage. We hear a lot about cold chain today and how things are distributed, and holding that material in a certain location that is GMP compliant. While Ashley, you had mentioned maybe there's changes in personnel, changes in leadership, keeps that super expensive, valuable material in a place where people can go back and tap into it in the future.
Yeah, we've seen that firsthand, and it's I think it's really important to have that continuity. It minimizes delays. I think it's hugely important, and sometimes not considered outright or in the beginning of walking into a relationship or partnership. Chad, did you want to comment at all? Did you have anything else?
Well, I have another quick question. I know that we're sort of coming towards the end of our session. But the one thing I kind of like to do anytime that we talk science or translational medicine is sort of give a look to the future. And I would really like to get each of your perspectives on what do you see for gene therapy over the next 5 or 10 years? I feel like we're at this inflection point where we're seeing a lot more development, a lot more research going in this area. But is there anything that sort of, I just want to get your overall vision of it. So if you could just weigh in on that, like, do you see it revolutionizing and now we're going to have, gene therapy for everything? Or is there some critical technology that you see emerging that will help us get over sort of the single therapy, single gene mutation type of approach?
Yeah, that's a fantastic question. I definitely think that the field is going to continue to evolve and grow. And I was talking to a colleague about this morning, because we tend to think of cell therapy and gene therapy obviously together. And it's, from our observation, it seems like we're not seeing quite as many cell therapies right now as we had expected. And some of that may be due to the operational facility availability at the site of administration to the patient to support some of those manipulations. But I think gene therapy is definitely more robust in some ways. And again, kind of lending to what Tom mentioned about cold chain and being able to have your material in a format that is usable for multiple patients, multiple sites around the globe. The fact that the agencies are starting to bring out more and more specific guidance documents to ocular gene therapy, I would say is definitely a good signal to the fact that they're seeing more and more of them being brought forward. So, I do expect that this area will continue to grow, certainly oligonucleotides as well. But, as much as this is very exciting, we also need to remember that on that one graph that I showed, there's still a very high percentage that are small molecules or other standard types of biologics. So we just have to be careful to not get a little bit distracted by the new tools and really think about the cost benefit ratio for the patients. If you can develop a therapy that meets that need and has that additional robustness or translatability, we definitely want to try to steer clients or manufacturers to those utilities as well, because, you get into gene therapies, they do get more and more expensive because of the narrow patient populations. And most companies do try to develop something that they can try to treat larger and larger patient populations with that single therapy versus creating multiple lines. So I think it's going to continue to evolve, and I'm excited to see what the next new breakthrough is. We're not done for sure.
Yeah, great point. We're just at the very, very leading and bleeding edge. And, want to just jump onto something that you said there about the patient communities. The advocacy organizations decades ago were really the ones championing and linking arms with the academic research institutions and the scientists. And a lot of that was and has still been in and around, rare indications. And there's a huge amount of gratitude. We've learned a lot because of that. And, as far as predictions go, I think that patient and advocacy connection with the research it's going to get even stronger. We see it now, the agency has done a tremendous job of putting patient first. The different forms that they're putting together for patients to express what is important to them, such that developers can take that, listen and learn. That's phenomenal. You see more advocacy organizations. FFB is a great example that they are championing science and granting, pathways for people to develop potential therapies and approaches, as well as taking it on the research themselves too. And that, I think, is something we'll see more and more of as these organizations get more savvy with regard to the process and understanding manufacturing and the regulatory requirements, that they're going to continue to take that on. And then I think what Leslie, you touched on too, is that we're taking what we're understanding and we're applying it to larger, more complex, beyond like a monogenic disorder, to have an even broader reach from a population perspective. And then I think too, we've had a lot of great success so far with viral vector delivery, but I do think that the non-viral approach is really going to be, turning a corner and coming of age in the near future.
Just Chad, what I'm seeing, I know we got to wrap up here, is we're involved in manufacturing GMP Cas9, and many people have heard of CRISPR-Cas9. And the idea of doing gene correction at the molecular level is just makes a lot of sense. It's easy to explain. People have a mutation. If you correct that, protein function would improve. And so we're super excited about CRISPR-Cas9. And we're in part have that excitement, I guess, because we're seeing what's happening with sickle cell disease, where there are people that are getting autologous gene therapy using gene editing tools, and people after 18 months have no transfusions when they've had pain for 34 years. And the NPR article is pretty famous out there, Victoria Gray. So gene editing, I think, is going to play a big role in curative, kind of corrective molecular gene therapy in the future.
All right, thank you.
All right, I think we are at the top of the hour, so we're at the end of our time. And I want to thank all of our speakers for joining us today. I really appreciate the time. A big thank you from FFB and Odylia, and from our audience. thank you for participating today. We do have a 4th webinar in this series coming up. We hope to announce that in the coming weeks for a final date and time. So stay tuned, sign up for any social media feeds that you usually use to get the latest updates from us on our next webinar. And we should post webinar number 3 in probably the next 2 weeks, usually when we get those up, right?
That's correct.
All right. All right, with that, thank you very much. And we will see you again soon.
Thank you.
All right. Thank you. Bye-bye.